Thank you so much for that introduction, Dr. McShane. I'm very excited to talk about our work today. This is a project that was born out of a postdocs project in the Kessler lab at Northwestern University, and it has now formed the bulk of my PhD dissertation work. I'll be talking to you about work that was published in Nature Communications recently about hippocampal adult-born neurons and ketamine's effects. Please let me know if at any point you can't hear me clearly. I am battling a cold right now, so if you can't hear me, just let me know and I will try to speak up a little bit more. Thanks. Okay. Moving right in, I am sure I don't have to remind you that depression has an incredible disease burden. And with its incredible prevalence, it's unfortunate that nearly a third of patients fail to respond to two or more antidepressant medications, thus forming the treatment-resistant group. For the patients who do respond to medication, there is still a delayed onset of therapeutic effects that lasts about 4 to 6 weeks. And the greater that time to response, the worse the outcomes tend to be and the higher the suicide risk. Naturally, this is a part of why there was such great excitement around the use of ketamine in antidepressant treatment, because it has effects that start within hours and last up to weeks. However, ketamine is not a miracle drug. As the group will be hearing about next week, it seems, there are a number of adverse events that are possible with the use of ketamine. And the mechanisms that govern those antidepressant effects versus the adverse effects are not very well teased out. What we do know is that ketamine's NMDA receptor antagonism is not sufficient for its antidepressant effects. In other words, other NMDA receptor antagonists don't have the same antidepressant profile as ketamine. In addition, we know that ketamine affects many regions of the brain, and there's been a lot of work on its molecular side, but there's little known about the specific populations and targets that govern its antidepressant effects. So based on that, I'd like to focus a little bit on the hippocampus, which is a region of the brain that is greatly affected in depression. As you know, it is a nexus of control for memory, emotion, and cognitive signaling, and it's also a unique site in the brain because it is one of 2 locations where there is ongoing neurogenesis, or the birth of new neurons throughout life in mammals. Specifically in the dentate gyrus, there is a multi-step process for neurogenesis that takes place, starting with neural stem cells, moving through neural stem cell proliferation, fate choice into the neural lineage, and maturation and integration into circuit. I'll just mention briefly here stage-specific markers that are expressed by cells at each of these different stages of maturation, with ASCL1 as a marker of proliferative progenitors, calretinin as a marker of immature neurons, immature dentate gyrus granule neurons, and calbindin as a marker of mature neurons, nu-N as a marker, of course, of the neuronal lineage in total. Specifically, adult-born immature neurons, marked here by calretinin and nu-N, for example, are electrophysiologically distinct. This means that they have a different activation threshold, they display different circuitry, they have unique characteristics compared to mature neurons. And it's been shown that blocking neurogenesis or ablating the entire neurogenic niche prevents the effects of first-line antidepressants as well as ketamine in mice. This is in part what motivated some of the recent work in our laboratory, which was published in 2019, showing that acutely activating this unique set of immature neurons without changing their number or even changing the niche induces a rapid antidepressant effect. After that observation, I then asked the question of whether activating these neurons could actually be what underlies ketamine's acute antidepressant effects. So moving right along into the paper that we just published a few months ago, I'll start with figure 1. I first established a paradigm in which a single injection of low-dose ketamine at 3 milligrams per kilogram in black 6 mice allowed for a pronounced behavioral effect at 24 hours post-injection. Before going into the data too much, I'd like to introduce the behavioral tests that I used throughout this study. The first of these is the social interaction test, otherwise known as the sociability test, and the readout for this is the sociability ratio, which is the amount of time that a test mouse spends interacting with a stranger mouse over the time it spends interacting with an inanimate object. Normally, mice prefer to be a little bit more social, so they do like to hang out with their new mouse friend. And stressed mice, however, will spend more time with the object. What I observed was that in ketamine treatment versus saline, ketamine tends to make mice more social. Now, in terms of novelty, a mouse tends to prefer meeting a new mouse over interacting with the mouse it already knows, and ketamine treatment also increases the preference for novelty. Finally, the tail suspension test is a marker of depression-like behavior in mice, and this is a measure of the amount of time that mice spend immobile, in other words, in a helpless, given up state, which is a marker of depression-like behavior, versus struggling, which indicates less depressive behavior. And ketamine decreases this phenotype. So once again, we've got the social interaction ratio, the novelty preference ratio, and both of these are markers of anxiety-like behavior. And then we have our depression-like behavior. At 24 hours, I established a clear phenotype. And then, using immunohistochemistry, I stained for immediate early genes that get turned on when neurons are active. And so I use them as a marker of neuronal activity. CFOS is one of these immediate early genes, and I observed that ketamine treatment greatly increases the expression of CFOS after 24 hours. I also validated this with a second activity marker, namely EGR1, or early gene response 1, which is another immediate early gene. To validate that these are in fact active neurons, I stained for NuN, our neuronal marker, and found that the increased activity takes place in the NuN positive group, namely neurons. To further delineate the maturity stages of the neurons that are activated, I stained for the maturity markers calbindin and calretinin, which I mentioned earlier. Calbindin is a marker of mature neurons, and calretinin is a marker of immature neurons. And I observed that the increased activity in ketamine is predominantly in the calbindin negative and calretinin positive groups, namely the immature dentate gyrus neurons. So after having observed this increase in activity, I wanted to then ask if this activity increase is necessary for ketamine's antidepressant effects. To answer that question, I used a powerful chemogenetic tool, namely DREADS, or designer receptors exclusively activated by designer drugs, which allowed us to specifically modulate the activity of immature neurons without altering the niche. This was a breakthrough in the field, and I'm excited to share with you how we used it. In my studies, I used two different DREAD types, namely the HM3DQ DREAD and the HM4DI DREAD. These are both modified human muscarinic receptors, and the DQ DREAD is an activating DREAD. It's tagged with the hemagglutinin tag, which allows me to stain for it by immunohistochemistry. I'll talk about that soon. And when activated by the synthetic ligand clozapine N-oxide, the DREAD HM3DQ leads to activation of the canonical GQ pathway, which leads to depolarization or activation of the neuron. Conversely, the HM4DI DREAD, when activated by CNO, leads to activation of the inhibitory GI pathway, leading to hyperpolarization or inhibition. So I first used the Cre-expressing mouse line, ASCL1 Cre HM4DI, to test whether inhibiting immature adult-born neurons would block the behavioral effects of ketamine. I expressed, again, this inhibitory DREAD receptor in ASCL1 expressing neural progenitors using a tamoxifen Cre system. ASCL1, as I mentioned before, is a marker of the proliferating progenitors, And over a period of 4 weeks, these ASCL1 positive cells mature into immature adult-born neurons at this stage that we're interested in. While these cells are maturing, I also expose the mice to unpredictable chronic mild stress, which is a paradigm of assorted stressors over a period of 3 weeks. On day 28, after this period of stress and when these cells have now reached the immature neuron stage, I injected the mice with our synthetic ligand, CNO, in order to activate the dread, in other words, inactivate the immature neurons. 2 hours later, I injected with ketamine. I maintained inactivation of these neurons using CNO in the mouse's drinking water. And then on day 29, 24 hours after ketamine injection, I subjected the mice to the same behavioral battery I discussed earlier. I observed once again some prominent behavioral results. From left to right, here are the results from the social interaction test, the social novelty test, again, tests of social and anxiety-like behavior, and the tail suspension test, which is for depression-like behavior, as well as the open field test, which is my locomotor control. In white, we have saline, and in green, we have ketamine, as before. In vehicle-treated mice, I'd like to point out that we observe the same increase in sociability and preference for novelty as before, as well as a decrease in time spent immobile, all depicting that ketamine decreases anxiety and depression-like behavior. However, in the second bars, with the CNO treatment, in other words, the inactivation of immature neurons, that behavioral effect of ketamine is not seen, all of which demonstrates that ketamine is necessary for this behavioral effect. Please also note that the mice showed no locomotor changes. To validate the inhibition of the immature neurons, I conducted immunohistochemical staining. The HA-tagged labeled dread-expressing immature neurons showed, the HA tag was for labeling the dread-expressing immature neurons, and there was no difference in the number of the total immature neurons across groups. However, there was an increase in activity after ketamine treatment that was suppressed by CNO. There was also no change in the total number of active cells across the groups. So having shown that inhibiting these neurons blocks ketamine's effects, I then asked the reverse, whether inducing immature neuron activity can mimic ketamine's behavioral effects. Here I'm skipping straight to figure 5 of the paper, just for brevity's sake. This time I use the ASCL1 CRE HM3DQ activating mouse line. In these mice, tamoxifen, as before, induced expression of the dread. The mice were exposed to stress once again, and then on day 28, mice were given a single dose of saline, ketamine, or CNO. CNO here activated the HM3DQ dread, leading to activation of the immature neurons. 24 hours later, mice underwent behavioral testing as well. Once again, there was no difference in locomotor activity. And very interestingly, we saw that ketamine and CNO both induce a prominent behavioral phenotype of similar magnitude in the sociability test, the novelty test, and in the tail suspension test. For further validation, I also conducted immunohistochemical staining and observed once again that there is no change in the total number of immature neurons. Ketamine and CNO both increase the activity of immature neurons, as expected with our CNO model, and there was no change, there was a great increase in the number of total active cells between ketamine and CNO, which makes sense because we increase the activity. All of this study in particular comes together to show that there is a specific cell population, namely these calretinin-positive, immature, adult-born dentate granule neurons, that's a mouthful, isn't it, that can be targeted to induce a ketamine-like and antidepressant effect in mice. Notably, this occurs without a change in the number of neurons it occurs without changing the neurogenic niche, and we showed not only that the activity is necessary, but also that increasing the activity of these neurons is sufficient for a ketamine-like effect. Now, we are still working on these ideas and questions, and as a continuing part of my PhD work, I am looking into how this activity of such a small cell population can cause such large behavioral changes, looking at cellular morphology, circuitry, and the molecular mechanisms behind it, as well as the mechanisms of sustained effects after a single ketamine dose, because as we all know, ketamine doesn't only have this acute effect, it also lasts for quite a while. And we have reasons to believe that the sustained effects, in fact, may have an additional mechanism. So these are the projects that I'm still involved in, and hopefully we'll be able to share the results with you soon. Thank you so much for having me, and I'd really like to thank everybody who has helped with this work. And now I believe it is time for questions.
Indeed. Fantastic. Thank you very much. I've learned a lot already, and I hope I'm going to learn some more. That's really, really exciting.
Thank you.
Just while others are coming up with their questions, we haven't had any yet in the Q&A. Can I just ask, I mean, I run an ECT clinic, and I'm always, you know, the students and doctors are always saying, how does ECT work? And one of the slides that I always show is a recent one looking at, I mean, we know that when you give rats ECT, then you get division of the cells in the dentate gyrus, the neurons in the dentate gyrus, and so that's one sort of explanation for why the hippocampus gets bigger with ECT. But there was a paper that came out in 2019 from a Japanese group that showed that the size of the hippocampus in remitters with ECT, it increased. It was increased at the end of the course of the ECT, and then it settled down again, and it decreased again 3 months later. So I always tell them, and I say it's a bit of a yarn, but it's a possibility that maybe there's something about this area of maybe there's something about the idea that the division of these neurons in the dentate gyrus actually is responsible both for the cognitive impairment of ECT, which of course parallels that time course that I've just said, gets worse towards the end of ECT and then better 3 months later. But also the effect. So I'm really excited to learn a bit more about this.
Those are very interesting observations. There's so much there. Yes, we have also done some work in our laboratory on ECT, showing, recapitulating what you've described as well about the increase in neurogenesis after ECT treatment. With most of these treatments, it has been found that after cessation, neurogenesis does normalize after a period of time. The duration of that time differs, of course, depending on the treatment. But I think it's interesting that you mention the reversed cognitive effect, because you may be aware there is quite a bit of research coming out now about the difference between the dorsal and ventral blades, or dorsal and ventral hippocampi.
No, I'm not. No, I'm just educate me.
Sure. The dorsal hippocampus has been most linked to cognitive behavior, while the ventral hippocampus, in part due to its proximity to the amygdala and to, frankly, all of the anxiety-based, fear-based structures, et cetera, is more linked to affective behaviors. So there's quite a bit coming out now about how those two poles of the hippocampus have differing effects. I would wonder about the effect of ECT and specifically the placement of the probes, for example, in affecting both of those aspects. I don't know too much about ECT, honestly, but I would be curious to find out if there are small differences in the location of the probe that might make a difference too.
I don't think they do. I think it's an overall effect. It's the electroconvulsive stimulation is a sort of equivalent to what happens in the human brain, which is doing the whole brain, not a sort of
Not focal.
no, not yeah.
Okay.
So I mean, did you see any difference in the effect in the dorsal and ventral hippocampus?
Yes. Unfortunately, I don't have my supplemental figures in my slide deck right now, but I can refer to the ideas for anyone who might have the paper pulled up. In supplemental figure 2, I believe it is, we do indeed look at the dorsal versus ventral the activity of immature neurons in the dorsal versus ventral hippocampus after ketamine treatment. My goodness,
Yeah.
excuse me, please. And for this experiment, sorry, I'm getting a little bit turned around here, but here we go. For the DI experiment, where we inhibited the activity of neurons, I also segregated my sections based on dorsal versus ventral hippocampus and quantified the activity of immature neurons in the dorsal versus ventral sections. And I observed that the increase in activity due to ketamine treatment primarily is in the ventral hippocampus, which is the part that, as I mentioned, is more associated with the affective behavior. Now, there's plenty of work to be done to follow up on that more, but it was interesting and in part reassuring that the ketamine effect is mostly on the affective pole at this acute stage, which matches a lot of the clinical data.
There's more that I want to ask, but we've got a question in the chat. Can you also relate your findings to Heather Cameron's previous paper on the antidepressant effect of ketamine in rats and how your findings differ from or relate to what she found? Do you know about those findings? And if you do, please tell us what they are.
Sure. So Heather Cameron's previous paper. Now, I do believe I know the paper, but if it's not the one that you mean, Dr. Denny, please do let me know. The one that I believe you're referring to is the one about how ketamine treatment increases the rate of proliferation of of maturation of neural progenitors. So for example, here, just speeding up this process.
Mm-hmm.
In other words, her paper was describing how neurogenesis in particular is not necessary for the acute effects of ketamine, but that instead the cells are just getting older faster. I think that, here's the link. Let's check it out and just make sure. Yes, here it is. Good. Good. I'm glad that my memory is not terrible. But yes, they mature rapidly in response to ketamine, but neurogenesis in particular may not be required. I think the main takeaway from this paper in relation to our work is that there are different stages of neuronal maturation. And even if you show that one of them may not be required for the effects of ketamine, there are many other stages. You cannot just show that taking away one cell population removes the effect. Secondly, showing that there is increased maturation does also may also play a role in the acute effects because you are increasing the let me pull up my diagram again, but if you're removing a population and then refilling, you're regenerating that neuron pool. Does that make sense? I'm not sure that was too clear. I'm sorry about that. I'd also like to point out that the Cameron paper is using the time point of 2 hours, and my studies are at 24. So when we say acute, it's very important for us to pay attention to the time points we're using, because at 2 hours, even though that is an acute effect, it's also a time point at which in mouse models as well as in humans, we tend to observe more of the acute hallucinogenic or more psychoactive behaviors. And so in the huge amount of behavioral testing that was required for us to identify our dose and time point, we made sure to find a paradigm in which we saw no changes in locomotion, no changes in rear ring behavior, no changes in things that might suggest those psychotropic effects. Namely, the 24-hour time point is a little longer than what the Cameron lab used. I hope that.
And they're looking at rats as well, which was a question I wanted to ask was, how do the two models differ, do you think? Or don't they? Are they comparable?
Mice versus rats?
Yeah.
So there is, let's see, obviously different organisms, obviously many differences there, but they are both rodents. The behavioral tests are a little bit different. The dosages, though, of ketamine that are given are similar, which is interesting. The vast majority of the work in both mice and rats for the subanesthetic doses of ketamine range from between 1 milligram per kilogram doses to up to 25 milligram per kilogram doses, both in mice and rats. So that's the extent of what I can share on that.
And you're on the low end of that at 3 milligrams per kilogram, aren't you?
Yes, because there is, out of all of the work that's been done, the higher doses tend to have more of the psychotropic effects that I just mentioned, for one, and also tend to have, the higher you go in dose, the less robust your antidepressant effects can be sometimes, which is very interesting to me. The lower doses can give you more of the antidepressant effect. In addition, the higher doses tend to change locomotion in the mouse and rat experiments, and it's a very important control that I wish more groups would do.
I think in the interest of time, but before moving to the more informal dialogue, I think there was another question from Professor Denny. If you don't mind me reading it to you. If you can discuss also the expression of ASCL1 outside of the hippocampus. Isn't there expression in the subventricular zone? And can you attribute some of your effects to other cells outside of the dentate gyrus?
That's a great question. With any of our inducible Cree models, it's always important to ask about off-target expression or off-target stimulation. And yes, the SVZ is also a site of neurogenesis. So things that are important for us to note are that we have validated this mouse model and we see very minimal expression of ASCL1 or our dreads outside of the hippocampus. And two, we've also validated a lot of this work using viral expression, namely with our ASCL1 Cree and then a viral expression into, or viral injection into the dentate gyrus specifically. And because we are able to do that targeted viral injection work, we can be sure about the location of our expression. So that's a long way of saying that's a very valid concern. We've validated this using another approach as well.